Mendel conducted a crossbreeding experiment between tall and dwarf pea plants to investigate the inheritance of a single gene. He gathered the seeds resulting from this cross and cultivated them to produce the first generation of hybrid plants. This generation is also known as the filial 1 progeny or F1. Mendel observed that all the F1 progeny plants exhibited the tall trait, resembling one of their parents, with none displaying the dwarf characteristic. Similar observations were made for other pairs of traits. Mendel noted that the F1 generation invariably mirrored one of the parental forms, while the trait from the other parent was absent. Subsequently, Mendel allowed the tall F1 plants to self-pollinate and was surprised to discover that in the filial 2 generation, some offspring manifested the dwarf trait. The characteristic absent in the F1 generation had now emerged. The dwarf plants constituted 1/4 of the F2 population, while 3/4 retained the tall stature. These tall and dwarf traits were identical to those of the parental types and exhibited no blending. Only one of the parental traits was expressed in the F1 generation, while at the F2 stage both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage.